Assalamualaikum warahmatullahi wabarakatuh. Good day for all of us. So we are from group 5. Today we will talk about enzyme. The first, we will talk about the history of enzyme. So in the late of 17th s until early 18th century, the digestion of means by stomach secretion and also the conversion of starch to the sugar by plants it and saliva were known. And after that, the next development, it is in the 19th century, where Louis Pasteur concluded the fermentation is catalyzed by the vital driving force present in the yeast cells that call as ferment. And after that, in the 1878, well, William Kuhne states the first name for enzyme. It is the Greek in the flour or yeast. And after in the 1897, where Edward Bashar states that sugar is fermented even if the yeast cells are not present in the mixture. So it is called the zymus. And after that, in the 1926, where James B. Summer states and success that in crashing the enzyme that called urasa, it is one of the pure protein. So the second, we will talk about the definitions of enzyme. We will take some opinions about this. The first is according to Kuhne in the 1878, where it states that enzyme comes from the word in plus zyme. This means that something in yes. And after that, according to Smith in the 1997, that states enzyme are biomolecules in the form of proteins that can function as catalysts. And after that, according to Christopher P. Austin, MD, 2020, states that the enzymes are biomolecules catalyzed, which are usually in the form of proteins, but can also in the RNA. And according to Suka, 2014, states that enzymes are complex protein molecules produced by living things and also work as catalyzed. And after that, according to you, the adult 2014 said that enzymes are the catalyst of choice, which is expected to reduce the impact of pollution and energy waste. And after that, according to Bersney 2003, each enzyme has a very precise and also different conformation as a result of several levels of protein structure. So, from some opinions, we can conclude that enzymes are biomolecules that function as catalysts in the chemical reaction. And after that, if this substance is not present in the process will be slowly and also almost all of the enzymes are proteins. The third, we will talk about the functions of enzymes. So the first is reduction. It is the additions of hydrogen, electron, and also release of oxygen. The second is dehydration. It is the release of water. And third is oxidation. So it is the reaction that release hydrogen molecule, electron, and also the additions of oxygen. The fourth, there is hydrolysis. It is the reactions of adding H2O or followed by the breakdown of molecule in the bonds of H2O. And after that, the fifth is deaminase. It is the reaction to release an amino group or NH2. And after that, the sixth is decarbonization. It's the reaction to release the CO2 and also carboxyl group. And there is the phosphorylation. It is the reaction that releases phosphate. After that, it is the catalyst that is one of the functions of enzyme. So there is the fourth is enzyme chemical arrangement. So there is the chemical arrangements of enzyme. According to Harden and Young in the 1905, enzymes or enzymes is the process of fermenting sugar into alcohol. The heat resistant part is called cosymase, and after that, many other enzymes also contain parts of protein, also products that are not protein. Not all of the enzyme are conclude as the protein. Next. Still in the enzyme chemical arrangement, the first part in the form of protein that is thermolabile or it is resistant to hot, it is called the isoenzyme. And after that, well, the part is not a protein, it is called the prosthetic group or the active part. Apoenzyme with prosthetic group, it is called holoenzyme. And after there are some in which of apoenzyme and also the prosthetic part are not united but separated. Also, there is the close relationship. And the loose prosthetic part we will call coenzyme. And next, we will talk about the enzymes in cell. So, cell can regulate or metabolic pathways by producing enzymes. So, enzymes are catalyzed that are more characteristic and also stronger than the metal ions and also other ungenic compounds. And after that, catalyzed are general in nature, only serve to speed up reaction, also that can be used over and over again. And enzymes are more specific, only used for one reaction. So inside the cell, the enzymes are not evenly distributed throughout the plasma. For example, is enzyme in the Calvin or also Krebs reaction are not found in the are found in mitochondria and also chloroplast. And after that, there are several enzymes found outside of the organelles, but they are also not distributed of the branching in the plasmic reticulum, uh, source from Fazia 2014.
Well, in this part, we're going to talk about the enzyme properties. So the enzyme properties of enzymes include the following. The first is enzymes are active in very small amounts. In biochemical reactions, only a small amount of enzyme is needed to convert a large number of substrates into products. And then the second is enzymes are not affected by the reactions, and then they catalyze under stable conditions. Under conditions that are considered non-optimal, an enzyme is a relatively unstable compound and is affected by the reaction it catalyzes. And then the third is enzymes do not affect the equilibrium of the reactions. So an enzyme will produce the equilibrium of the reaction at a higher rate. And then the last is the specific enzyme catalyzed action, because the enzymes exhibit specificity for the reactions they catalyze. It's like an enzyme that catalyzes one reaction will not catalyze another. Next. So in this part, we're going to talk about the enzyme nomenclature. Well, the naming of enzymes is trivial and systematic. For the trivial, is classified according to the first is the substrate catalysis. And then this enzyme names end in acid. And then the second is the product form, and then the third is the substrate and the type of reaction mechanism, and then the fourth is the product synthesized, and then the last is the general type of reaction catalyzed by enzymes. But for the systematic nomenclature, it's according to enzyme commission or EC. Next. So for the trivial system, the first is according to about the enzymes based on the substrate catalyzed. This part is named N in AC except for some proteolytic that end in. The example is maltase from maltose and pepsin from peptone. So this picture is of the lactose breakdown reaction by lactase enzyme. Next. And then the second is the enzymes based on products formed due to enzyme activity. It's based on the products formed due to enzyme activity. Example is like the citrate synthase with the reaction product from citrate. This picture is of the citrate formation reaction by citrate synthase enzyme. Next. And then the third of the trivial system is the first is enzymes based on products formed due to enzyme activity. Example is the pyruvate compounds that can be converted to acetyl CoA by oxidative decarboxylation reactions and to oxaloacetate by dehydrogenases. And then the fourth enzyme based on the products formed due to enzyme activity. Example is like the citrate synthase, which catalyzes the condensation of acetyl CoA and oxaloacetate to produce citrate. And the last is based on the type or nature of reaction catalyzed that without a specific substrate. Example is like the transfer enzymes, hydrolysis, synthesis, and the enzymes that add functional groups or double bonds. Next. So for the systematic nomenclature is based on the following principles. So it's like the first is the first digit indicates enzyme class, and then the second digit is a subclass, and then the third digit is detail the enzyme subclass, and then the fourth digit is usually a list number given by the enzyme commission. Example, it's like enzyme commission 211 methyl transferase or transfer CH3. And then after we talk about the enzyme, we're going to talk about the enzyme classification table based on the reaction type. The first is the class 1, is the enzyme class is oxidoreductase. This reaction type is oxidation reduction reactions or transfer or electrons from one substrate to another. And then the enzyme class 2 is transferase. This reaction type is transfer of atoms or groups from one substrate to another. And then the enzyme class 3 is hydrolysis. This reaction type is hydrolysis reaction or breaking bonds in the presence of water. And then the fourth enzyme class is lyase. This is addition of a functional group to a double bond or addition or cleavage of double bonds with the release of the function groups. And then the fifth of the enzyme class is isomerase. This is the isomerization reaction or rearrangement of substrate atoms. And then the last is the sixth enzyme class about the ligase. This reaction type is formation of CC or CS or CO and the CN bonds. This followed by cleavage of the isophosphate from ATP. And then after that, there is the enzyme subclass classification table. From the class oxidoreductase, this is the subclass is oxidase, that the type of the reaction catalyzed is substrate oxidation. And from the subclass reductase is substrate reduction. And from the subclass dehydrogenase, the release of hydrogen atom or H2, the double bond formation. And from the class transferase, this subclass is 2 is transferase, the transfer of amino groups between substrates. And then the second is kinase. This is the transfer of phosphate groups between substrates. And then the next is about the hydrolysis class. The subclass is lipase, that hydrolysis of ester groups in lipids, and then this is protease, that hydrolysis of amine groups in proteins, and then the nuclease, that hydrolysis of phosphate groups in nucleic acids. And then the class of lyase, the subclass is dehydration, that type of reaction is water loss. And then the next, the decarboxylase, that the type of reaction is loss of carbon dioxide. And then for the class isomerase, the subclass is epimerase. This is the isomerization at the chiral center of the substrate. And then the last class is ligase. The subclass is the first is synthesis, the formation of the new bonds involving ATP. And then the third and then the second is the carboxylase, the formation of new bonds between substrate and CO2 involving ATP.
Okay, so this is the 3D structure of the enzyme substrate complex. After we know the history of enzyme discovery until its classification, then we must also know how enzymes work. Enzymes have an active site that completes certain parts of the substrate. The enzyme can be considered as a lock that will bind to the substrate. So substrate is considered as the key.
All of these are known as the lock and key theory. This theory was proposed by Emil Fischer in 1894. If the key is inserted into the unsuitable padlock, then the padlock does not open. This is one of the way enzymes work. Look at this one. Enzyme has an active site that will bind to a substrate appropriately and will produce a product. You should know that the binding of substrate or some other molecules to an enzyme causes a change in the shape of the enzyme. It is known as induced fit theory. Even the lock and key theory, the substrate must have the same shape as the active site. Then this is different in this theory. There is a change in shape experienced by the active site. So this theory is more accurate description of the enzyme action. This was proposed by Daniel Kosland in 1958. Look at this. The active site and substrate has a different shape but can be linked. Why did it happen? It is said that the enzyme is flexible, so the active site changes shape to wrap around the substrate. If we talk about enzymes, there's a cofactor. Cofactor can be considered as the helper molecules. This is a substance that increases the enzyme activity. The cofactor is divided into three, namely prosthetic groups, coenzyme, and metal ions are also known as enzyme activators. The prosthetic groups are thermostable organic compounds that closely bond with apoenzyme. Example is heme and biotin. Whereas coenzyme are also organic compounds but are loosely bound to the enzyme. Example is NADP. Then, metal ions, different from others, are inorganic compounds that temporarily bind to the enzyme active site. Example is zinc. Look at this one. Cofactors will bind to active site to make it easier for substrates of different shapes to bind to the active site. We can say that cofactor can affect enzyme activity. Then, what else can also affect the enzymes? Their temperatures, pH, concentration of enzyme substrate, and inhibitors. The first is temperature. The optimum temperature for enzyme is 40 degrees Celsius. Too high temperature will cause protein denaturation. But if the temperature is low, there are few collisions between enzyme and substrate. The second is pH. There are different optimum pH depending on the type of enzyme, but commonly pH is 7. It is neutral. Changes in pH can affect changes in the amino acid on the active site. Look at this one. Some enzymes have a very acidic optimum pH and some are very alkaline. Pepsin has a pH of 2 in acidic state, while the amylase enzyme has a 7.5 pH, it is alkaline. The third is concentration of enzyme substrate. Rate of the enzyme catalyzed reaction increases in the proportion to the concentration of enzyme substrate until the reaction reach the maximum rate. Look at this one. After reaching the maximum point, then the reaction rate runs constantly. The last is inhibitors. There are two kinds of inhibitors. The first is competitive, the second is non-competitive. What's different with both? The competitive one works simultaneously while the non-competitive works permanently. Look at the non-competitive one. The non-competitive one does not stick to the active site, but in the other part. This does not block the substrate, but changes the shape of the active site, so the substrate cannot stick. While the competitive inhibitor has similarity of structure to the substrate, so and stick right on the active site. It simply blocks the substrate without changing the enzyme. So that's all if we talk about the enzyme. We hope you can understand more after watch this video. Thank you.